This lesson is proudly brought to you by Kagiso Trust in partnership with the Limpopo Department of Education and Capricorn FM. Kagiso Trust, overcoming poverty.
Class is in session.
Ding, 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 ding.
We don't need an air horn for it. Straight. All right, 7 minutes after 5. We are joined here today by Mr. Majia, and he's back again for the second time. To come give us lessons on life sciences, term 1, and the discussion for today will be mainly focused on meiosis. Welcome back.
No, thanks very much.
You're a regular now, you know.
Yeah.
We will speak to the Department of Education to secure you a permanent slot here. All right, thank you so much. Mr. Majia is here to take over, of course, Life Sciences Term 1. Now, if you've got any questions, feel free to send them through right now to our WhatsApp number, 082-654-2446. Over to you.
I think also before he starts, he can just sort of refresh our memory as to where he's from and which school he teaches. Yeah,
definitely. I'm Mr. Majia SR, attached to Texila Secondary School. And then I'm sure we're going to enjoy this lesson.
Definitely.
Thank you.
All right. Over to you, Sam.
Okay. Thank you. Good evening, my lovely students. I'm sure my principal is also listening, because today when I went to school to fetch something. He also said, no, remember, I'm also listening. It's Mr. Amara. Okay.
All right.
No, thanks. My students, last week, or last session, we made it a point that at least I emphasized something to say. I'm going to coin a particular question based on DNA, although today we're going to continue with meiosis. So before we continue with meiosis, I want you to write the following question down, because normally this is the part that challenges a lot of learners. So you may write this statement down. It says, the diagram below shows the sequence of nitrogen bases. The diagram below shows the sequence of nitrogen bases of a strand of a DNA. The diagram below shows the sequence of nitrogen bases of a strand of DNA, which codes for a part of protein molecule. And then down there, they give you the triplet of bases. They say GTC, and then put dash, and also write TTG, and then put dash, and write AGT. I said GTC, TTG, and then AGT. And then again, there's a statement that follows that triplet of bases. It says, from the DNA sequence above, write down the mRNA codon sequence that reads from left to right, and then it's 3 marks. Remember, last time I told you that 7 plus 3 is equal to 10, 9 plus 1 is equal to 10, 5 plus 5 is equal to 10. That what we're teaching is this 10. So I want to guide you with regard to the approach that we use in order to make the triplet of bases. So the very same approach that I'm teaching you right now, you are going to apply it to each and every question that you are going to come across. As long as you are not going to cram, that's how I'm going to take my time here. So now, before you answer this type of question, make it a point that you draw a table and demarcate it into 3 columns. Then the first part, just write DNA. And then the second column, write mRNA. Remember, mRNA is formed from the DNA. Just write mRNA. And then the third column, write tRNA. DNA. And then tRNA as the second column. mRNA as the second column. And then the last one, tRNA. And then again, remember, we have not yet started to answer this question. This is the strategy that we are going to use for each and every type of question that needs you to write the triplet of bases. And then now also write DNA, just write DNA. And then below DNA, remember we talked about ATCG as a formula, meaning that adenine pairs with T, which is thymine, and then C pairs with guanine. Just write ATCG. And then also write RNA, just below RNA, write A-U, and then below A, write C, and then write dash and G. So this is the approach that we are going to use. Remember, you are going to have a table which has got DNA as the first column, mRNA as the second column, tRNA as the second column, and the third column, and then also you end up having ATCG under DNA, and then AUCG under RNA. Now, let us check how you are going to approach this type of question. Remember, it says the diagram below shows the sequence of nitrogen bases of a strand of DNA, which codes for a part of protein molecule. Then they said GTC dash, then TTG dash, and then AGT. So now, and then from there, it says from the DNA sequence above, write down the mRNA codon sequence that reads from left to right. Examiner is forced to tell you something about the triplet row basis. Number one, he must tell you that this triplet row basis, they fall under mRNA or DNA or tRNA. Now let us check this triplet row basis. They fall under, they said it's the triplet row basis of mRNA. So now we are going to write mRNA under, you are going to write this triplet row basis. And so let me check. The diagram below shows the sequence of nitrogen bases of a strand of DNA, which codes for, this is the DNA. Now you are going to write, remember you've got your table now, you are going to write the GTC under the DNA, and then just below GTC you also write TTG. Just below TTG you are going to write AGT. Why? Because they said these are the triplet row bases of DNA. Then you write DNA, those triplet row bases under the appropriate column. Now, you are going to check as to whether maybe they want the triplet row basis of tRNA or mRNA. Let us check the statement. It says, from the DNA sequence above, referring to GTC, TTG, and AGT, write down the mRNA codon. Now they want the mRNA codon. That is from left to right. Now let us check what is it that we are going to do. You are going to refer from the keys that I gave you, and then remember we've got DNA, mRNA, and then tRNA. Now because they want the mRNA from the DNA, we are going to do it like this. If we check G, because we want the mRNA, if we omit that M is RNA, and then we're going to use AUCG. Let us check now. Then we check G, it complements with, you go back and check from the key. G complements with C. And then you also check A. Remember, we're checking under RNA. That A complements with U. I'm sorry, that A. Then you are going to check, because the first one is G. And then T, you check under RNA, there's no T, then you go back to DNA. Then wherever you see T, then it complements with A. Then you write A. And then from there, the other one, you check C. Then you check again. C always pairs with G. Then you're going to write G. Then you are going to end up having CAG. Then let us check TTG now. For TTG, you check T with A, because under mRNA is not there. Then you check T pairs with A, and then you write another A again for that other T, and then for G you write C. So you are going to end up having AAC. And then the last one is AGT. Then A, you check from the diagram from mRNA, because the question is about mRNA. Many learners make a mistake to say A pairs with T. Here they're not talking about the DNA, they're talking about the mRNA. So we are going to say A, if we check under mRNA, the one that complements mRNA is U, that complements A is U, and then G, you write C, the one that complements T, then you check from the diagram under DNA, then the T complements with A, then we are going to end up having UCA. So this is the approach that we are going to use. And please, if ever you still don't understand this part, although I know I explained it thoroughly, make it a point that you go through different questions. And the next time when we meet, we are going to discuss a lot of things, because the chapter that we are going to discuss is very short. So at least some of the questions, although you are going to pose them here, some of them we are going to discuss them during the next lesson. So thank you. Now with mitosis and meiosis, remember, it covers almost 23 marks. It's 11 marks for paper 1 and also 12 marks for paper 2. So I'm just going to explain the peripheral explanation based on mitosis because you did it in grade 10. Then it's going to form part of our introduction, and then or part of our lesson. But before that, because we're going to deal with chromosomes, you must know what is a chromosome. Wherever you are, or if you're not having a pair of scissors, but you know a pair of scissors, it has got two blades that are joined together by something, a round-like structure. So you are going to equate that with the chromosome. Those two blades, each blade is like a chromatid, another blade is like another chromatid, and then that round-like structure that joins it at the center, that joins the two blades at the center, is called the centromere. So in other words, you are going to have the chromatid, two chromatids held together by the centromere, then they form a chromosome. And then the other point that I want to bring forth is that where do we find these chromosomes? Remember, we've got a human being is made up of millions and millions or even billions of cells. Why? Because in grade 10 you learn that many cells form the tissues, many tissues form organ. Many organs form a system, and then many systems form an organism. So you are an organism that is formed by many systems, and then those many systems are formed by many organs formed by many tissues, and then many tissues formed by the cell. So in one cell, we've got, I'm talking about cell of a human being because we've got different types of cells, like for argument's sake, the cell of a rat has got 42 chromosomes, whereas the cell of a dog has got 78 chromosomes. But now, for your syllabus, we're going to dwell much on the cell of a human being. Remember, it has got 46 chromosomes. Now, how are these chromosomes arranged? They are arranged in the form of, they call it a karyotype. So maybe before I define the karyotype, let me give you this example so that you understand it better. If in a classroom situation, I start to arrange you accordingly, maybe I say, you need to sit according to the way I'm going to instruct you. Let me say, I say, Tolly should sit with a tall boy or a girl, and then fatty to fatty, slender to slender. I'm arranging you according to size and shapes. So it is like this scenario. When we talk about chromosomes in a cell, so remember I said there are 46, but they are arranged in pairs, meaning that we are going to have 23 pairs. So immediately we talk about, and these chromosomes are arranged according to their size and shapes. Then the arrangement of chromosomes in a cell according to size and shapes, that is what we call the karyotype. And then this karyotype, remember I said each chromosome has got 23 pairs, meaning that it has got 46 chromosomes. Now, pair number 1 up to number 22, there is a collective name of those type of chromosomes. They are called the autosomes or body chromosomes. And then we also have pair 23, which is called the sex chromosomes. In some books, they talk of the gonosomes. And then sometimes when you check pair 23, you realize that pair 23 has got XY chromosomes. If it is XY, know that this karyotype is the karyotype of a male. And then if we've got XX, or pair 23 is XX, just know that this is the karyotype of a female. It is very vital for you to know this, because when we'll be busy with genetics next time, when we talk about sex-linked disorders, we're going to use this. Then the other point that I want to bring forth is that you need to familiarize yourself with the different types of sets. Number one, if we've got one set, a set is a ploid meaning set. So if it is one set, just write 1 or one set. In brackets, just write N. And then write 23 chromosomes. And also write the equal sign. Just in front of that equal sign, write haploid. In other words, one set is called the haploid. And then if we've got 2 sets, you write 2 sets. In brackets, write 2N. And then also write 46 chromosomes. Remember, we're talking about the cell of a human being. And then write diploid, di meaning two. In other words, we've got two sets. And then sometimes we end up having 3 sets. Just write 3 sets. In brackets, write 3n. And then almost 69 chromosomes. And then write the equal sign and write triploid, tri meaning 3. In other words, we've got 3 sets. And then the fourth one, write 4 sets. In brackets, write 4n. And then 92 chromosomes. And equal sign. And write tetraploid, T-E-T-R-A, and ploid. Now, remember, when you talk about triploid and tetraploid, because there are more than 2, this are more than 2 sets, then we regard them collectively as the polyploid, and poly meaning male. And then normally this happens during abnormal meiosis. So these are the things that we are supposed to take into cognizance. Now, with regard to mitosis, I'm just going to give the peripheral explanation about mitosis, because I know it's a revision you did in grade 10, but it's going to form an integral part for us here in grade 12 with regard to meiosis. And then maybe to highlight you with regard to that one, the main reason is that meiosis, remember, it has got meiosis 1 and meiosis 2. So meiosis 2 is similar to mitosis. That's why it's very vital for us to know or to revise something about mitosis. So with mitosis, it has got 5 phases, but mitosis and meiosis, just know that these are cell division, is when the cell divides to form 2 or more than 2. Now let us check the formula that we're going to use to know all those phases of meiosis. You may write this mnemonic device or the abbreviation IPMAT. IPMAT. And then I for interphase, and then P for prophase, M for metaphase, A for anaphase, and then T for telophase. Now from here, if you check all these 5 phases, which one is at the center? Obviously it's metaphase. And then the most important thing that is interesting with regard to metaphase, I'm going to start it from metaphase. Meta means middle, and then it's derived from Greek word meaning middle. So in other words, with metaphase, I'm just going to give you the icons of interphase, prophase, metaphase, anaphase, and telophase. And then with metaphase, because meta means middle, as long as chromosomes are along equator, then you need to know that, oh, this is metaphase, because meta means middle. And then after metaphase, we're going to have the phase that comes immediately after metaphase, which is anaphase. And then here, just know that something is going to move to the poles. From equator, something should move to the poles. The question is it chromatids or chromosomes? So now with mitosis, under metaphase, we must have the single chromosomes. We are going to have single chromosomes that arrange themselves along equator. They don't arrange themselves as homologous. It's single, one on the left-hand side, maybe the other one on the right-hand side. And then now, with anaphase, like I said, something is going to move to the pole. The question is, what is it that is going to move to the pole? Remember, I said the chromosomes is made up of 2 chromatids held together by the centromere. So here the centromere divides, and when it divides, the chromatids now, they're going to move to the opposite poles. In other words, it is not the old chromosome that move to the opposite pole. It's just the chromatid. And then with telophase, then how are you going to know something about anaphase? As long as you see that chromatids are moving towards the poles, then you know that this is anaphase, which is the phase that comes immediately after metaphase. Then how are you going to, what are the icons that are going to guide you about the telophase? And then here, because we said mitosis is the cell division, is when the cell divides. So now, when the cell is about to, or as constricted, just about to divide into 2, then you know that this is the last phase, which is called telophase. Then let us go back to interphase and prophase. And then interphase is just the preparator stage where the DNA replication takes place. And then what are the icons that you are going to see there? You are going to see the chromatin network, not chromosomes, the chromatin network. And then with prophase, pro means first. And then meaning that here, This is the first phase where chromosomes are visible. That is all about the icon that we are going to have with regard to mitosis. And then from there, you are supposed to know something about the definition of meiosis before we dwell much on the whole process. And then when you check meiosis, when you define meiosis, you must have the following 4 concepts. Number 1, you must know that it is a cell division. Remember I said mitosis and meiosis are cell division. So now we are going to say meiosis, the cell division, you get a mark for that. Whereby a diploid cell, you also get a mark for diploid cell, or somatic cell undergoes 2 cell divisions, then you get another mark for 2 cell divisions. And then to form 4 dissimilar, different, and then I'm going to explain thoroughly what brings forth this difference. And then, because we're talking about dissimilar haploid cells, and then you're also going to get another mark for haploid cell. And then those haploid cells are regarded as sex cells, and then sex cells, in brackets, we may write gametes. Remember, when we talk about gametes, we're talking about the sperm cells and also the egg cells. In other words, I'm preparing you for the chapter that we're going to discuss next time with regard to, which is reproduction. And then now, under this, we're going to have gametogenesis, spermatogenesis, and also oogenesis. Then gametogenesis, from the word gamete, it embraces both parts, that is spermatogenesis and oogenesis. And then when you come back from break, and then I'm going to guide you with regard to their definition. Then from there, we're going to continue with the other section. For now, let us go for break. Thank you.
Right, it's 28 minutes, 28 minutes after 5. Capricorn FM, Limpopo Department frequency. Meiosis, it's in it, class. Kamagalo Namalali. Going back to school, are you?
Yeah, man. I don't even know what he's talking about, to be honest.
You didn't do life sciences at school?
No, I did accounting.
Oh,
commercial studies.
You're a numbers guy.
Yeah. Tell me about numbers.
All right, Mr. Majia, we'll be continuing in a short while. With Nhlamulo Marule, and then we're going to be getting into your sports updates with Kamarelo Namalani.
Taking out insurance is easy, but understanding the terms and conditions of your cover can be complicated. Let us help you. Tune into Capricorn FM Talk every second Wednesday at 7 p.m. from February to July 2020 and listen to insurance experts explain the standard clauses of an insurance policy contract, and you could win 250 rands worth of airtime. T's and C's apply. Understanding my insurance policy, brought to you by the South African Insurance Association.
News headlines on Capricorn FM.
Let's look at our headlines now. The Department of Social Development in Limpopo is calling for a harsh sentence to be meted out against perpetrators involved in the gruesome murder of 14-year-old Morongwa Marakalala at Silvermine Village. The NPA says they have made arrangements allowing for a presiding officer from another court to preside over the case of prosecutor Rachirumela Leonard Makado, who is based in the Thohoyandou Magistrates Court. And AU Chairperson President Cyril Ramaphosa has emphasized the need for global solidarity, calling on world leaders to continue working together towards equitable access to medical equipment, technologies, and best practices to combat COVID-19. Your final bulletin at 6 p.m.
Capricorn FM Sports, on and off the field. We've got you covered.
Now for your final look at your sports for today. Golden Arrows assistant, the coach Amandla Gugazi, says Kagiso Trust's passing is a big loss to the football fraternity in the country. The defender's passing was confirmed by the club this morning, and his family has confirmed that he is a heart failure. Ngwengwe made his debut for Arrows in the August 2013 and went on to play more than 140 matches over the years. Gugazi says everyone at the club was devastated by the sad news they received today.
We were supposed to start our Zoom session at 9. Unfortunately, when we got the news, players were devastated, everybody was emotional, we couldn't handle the session. It's a big loss into the team, big loss into the family of Golden Arrows.
And with the country under lockdown and with not more than 50 people allowed to funeral gatherings, Ngakazi says they will find a way to pay their last respect to their beloved player.
We will find a way to pay our respect to his family. Maybe today we will go to his family to see what really happened. It is our obligation as coaches and technical team so that we don't compromise the current situation that is in the country.
And now, meanwhile, in rugby, Eastern Province Rugby Union Deputy President Bantwini Matika tendered his resignation today amid allegations of gross misconduct leveled at him by the union. Matika and two other EPRU officials, Vice President Eldrick Chito February and consultant John Shippers, are being investigated for allegedly attempting to defraud the provincial union of over 450,000 rands. Both Matika and February were influential in ousting former EP President Chiki Watson as part of the Transformation Rugby Coalition, and were subsequently elected to serve on the board of the amateur union along with the current president Ander Remban in 2017. Matika opted to step down with immediate effect after the investigation, and of course it's still ongoing right now, and the accused could face the criminal charges. Retired Justice Alex Mbati was last week appointed to lead the investigation. And now finally, in the English Premier League, Manchester City winger Raheem Sterling has cast a doubt over the Premier League's plan to resume matches next month, with many top stars still not back in training. The league is anxiously keeping its fingers crossed, hoping that Germany's experiment in biosecure football works, and the richest football league in the world hopes to relaunch its own competition sometime in June. Whether that is desirable or feasible has been the subject of fierce debate so far. If Project Restart is given the go-ahead this week, players are expected to begin non-contact training together, but the planning process has been troubled with difficulty. Some players have voiced a deep reservation over their safety, and there's also concern that after a long and unusual layoff, players have no hope of reaching proper match fitness before June, and a flood of summer injuries might therefore accompany the 92 fixture schedule for empty stadiums during a hectic 7-week period. Sterling has insisted players would require at least a month of full training before they can be pitched back into the big match.
I know the clubs are in talks and, doing their best that they can to try and find the best solution for this. But yeah, we've had a few meetings cancelled, even on Zoom to talk about the restart. We have to just wait and see, really. You can't come back in with 1.5 weeks, 2 weeks. You need a full 4 or 5 weeks. Especially if you're going to go back into competition where it's not you're playing for friendly, playing to literally win, and it's going to count for something. So you do need that preparation, definitely.
And this is all for your sports for today. I'll be back with more tomorrow morning on the Go Breakfast with Complexion and Mpoma Shita. For more updates, follow at Capricorn FM News on Twitter. And for Capricorn FM Sports, I am Kamarelo Namalani.
Keeping you up to date with the latest sports news. Capricorn FM Sports. Rooted in Limpopo's cultural diversity, we are Capricorn FM, Limpopo's hottest frequency.
This lesson is proudly brought to you by Kagiso Trust in partnership with the Limpopo Department of Education and Capricorn FM. Kagiso Trust, overcoming poverty.
25 minutes heading to 6. Rosanale, Mr. Maji. Rosanale Baba Libija Minier, Skolo.
I'm a teacher.
Hey, what teacher? Hey, na go jail. Menier Maji. All right, now Mr. Maji in the studio by Teacher Maji, Babulation Al Life Sciences today, term 1, and the topic is Meiosis.
Okay, thank you. And then remember, before we went for break, I was talking about gametogenesis and also oogenesis and spermatogenesis. Remember, gametogenesis, the compound weight made up of gamete and genesis. Genesis meaning the formation, and then gameto from the word gamete. In other words, the formation of gametes. And then spermatogenesis. Genesis again means formation, and then sperm cells, or is the formation of sperm cells. And then oogenesis from the word ovum, that is the formation of the egg cells. We are going to deal much with regard to this one. One will be discussing reproduction, just the introduction part of it, because the two chapters are linked together. And then the other point that I want to bring forth is that you need to know the position or the places where this meiosis takes place, because sometimes the examiner is going to ask you questions based on that. He may say, name the places where meiosis takes place in plants. So in plants, you are going to say male. In males take place in the anther, and then females take place in the ovary. And then in animals, you are going to talk about 2 places again, from males and females. And then in males, it takes place in the testis, and then in females takes place in the ovary. But the other thing that you need to take into cognizance is about the importance of meiosis. Number one, it produces gametes. When you talk about the sperm cells and also the egg cells, the main type of cell division that play a major role is meiosis. Besides meiosis, meaning that sperm cells cannot be produced and then egg cells cannot be produced. Although I said next time when we be discussing reproduction, I'm going to tell you a lot about the spermatogenesis, the formation of sperm cells, and also the oogenesis, the formation of egg cells. How are they formed? And then the other point is that it halves the chromosomes number from diploid to haploid. And then why diploid to haploid? Remember, diploid I said is 2N or 2 sets, then haploid is 1 set. The main reason why it's supposed to be reduced to, it halves the number of chromosomes from diploid to haploid is that the sperm cell, remember we said the normal cell should have 46 chromosomes. So now if we have 46 chromosomes and we take half of 46 from the sperm cell, then we end up having 23. And then from the egg cell again, 46 end up having 23. Then it's half. And then this 23 plus 23, when they fuse together, they form 46. Now the cell is normal. That's why it plays a major role with regard to the halving effect. Although, don't worry, we will deal much about this in reproduction. And then the third one is it introduces genetic variation. And then remember, variation is brought forth or comes across, is brought forth with regard to two processes. The first one is crossing over. The other one is random arrangement of chromosomes along equator. Now, with meiosis 1 and meiosis 2, the most important thing that I want to bring forth is that I said meiosis 2 is similar to mitosis. So in other words, we're no longer going to discuss meiosis 2 because I said it's similar to mitosis. Like for argument's sake, when you talk about metaphase, just to give you clarity with regard to this one, remember during mitosis we said during metaphase, single chromosomes are arranged along equator. Even during meiosis 2, which is metaphase 2, then the single chromosomes are arranged along equator. And then when you talk about anaphase, under mitosis, remember we said that the chromatids move to the opposite poles. And then under the anaphase 2, the one that I said is similar to mitosis, we're also going to talk about the chromatids. We're going to say chromatids move to the opposite poles. So that's why I said they're similar. Then it will be repetition. But now let us check meiosis 1. How is it special? It is special because it differs from mitosis. It also differs from meiosis 2. Let us check how does it differ from those. Then we start with prophase 1. By why prophase 1, not starting with the DNA? I mean, with not the interface. Maybe let me guide you with regard to the interface. Or let us just move on to with prophase 1, and then when we're done, maybe I'll give you the summary towards the end. But with prophase 1, number one, you must know that here it differs from the other prophase of mitosis and even meiosis 2 with regard to, in the sense that it has got what we call the crossing over. Remember about crossing over, you may use your two hands, check the finger that is next, two fingers that are next to the thumb, meaning that the finger at the center and then the other one that is next to the thumb. Then you are going to use both of them, the one on the left hand side and also the right hand side. So let me say the middle finger is like, and the middle finger and this finger that's next to the thumb, they are like chromatids, two chromatids. Then you are going to say, number one, homologous chromosomes move close to each other. Then that's the first point. You are going to get a mark. Remember this homologous chromosome, the other one is coming from the mother, the other one from the father. Why homologous chromosome? Homo means the same. They are of the same size. And then the two middle fingers, or the two fingers, you are going to cross them, like you are doing an X. Then you are going to say chromatids overlap. You get a mark for that. And remember, this point of overlapping is called the chiasma. Although some books, they talk of chiasmata. Chiasmata is the plural form of chiasma. So you need to write chiasma, and then you have got 2 points now. Then now you're going to say these chromatids are exchanging genetic material. You get the 3rd point. And then the fact that they're exchanging genetic material, this brings forth or introduces genetic variation, which is the 4th point. And then ultimately, variation takes place. Then this is about prophase 1. The other point that you need to know under metaphase 1 is about the chromosomes. Now, remember, during metaphase, under mitosis, we said single chromosomes are arranged along equator. But here, the chromosomes are arranged in homologous pairs. And then, in other words, for argument's sake, we are going to have 2 this side, 2 the other side. So, meaning that we are going to have like 4 chromosomes, 2 on the left-hand side, 2 on the right-hand side. They are arranged according to homologous pairs. Then now, with anaphase, remember I told you that with anaphase, something is going to move to the poles. So the question in this regard now is, what is it that is going to move the poles? Now, because we've got 2 chromosomes at the center, we've got 2 on the left-hand side, 2 on the right-hand side, there's no need of dividing 1 chromosome. Then the whole chromosome now, let me say we've got 2 on the left-hand side and the 2 on the right-hand side, the upper ones and the lower ones. The upper ones are going to move towards this opposite pole, and then the lower ones are going to move towards the opposite pole again. So in other words, now here, the whole chromosome moves to the pole, not chromatids. Chromatids only move when we have single chromosomes along equator. Now, because we have got homologous, that's why the whole chromosome moves to the opposite poles. And then telophase 1, in conclusion, you are going to say the chromosomes reach the poles of the cells. And then point number 2, each pole has half the number of chromosomes present in the original cell. And then e.g., if original cell was having 4, then half of 4 is 2. Then the cell membrane is going to constrict and divide the cytoplasm, and then to form 2 cells. Now, on that note, you need to know something about questions that can be asked based on this. Madam B, before that, I see we still have time. I'm going to, during the last 10 minutes, I'm going to coin a lot of questions based on meiosis. But now, sometimes examiner is going to request you to compare mitosis and meiosis. So I don't want you to cram this. I want you to, we're going to do it together. Wherever you are, make it a point that you have a blank page, and then this blank page, draw a circle at the center, and then that circle you may write nucleus, label it the nucleus, and then the space outside that nucleus, you may say it's the cytoplasm. Then now cut that page into 2 pieces. And then now when you cut that into 2 pieces, obviously one, the cytoplasm divides, and then the other point, the nucleus divides. So, and then sometimes the division of cytoplasm is called the cytokinesis, and then division of nucleus is called karyokinesis. So, because sometimes examiner is going to say, what is the division of nucleus? You must know that that is karyokinesis. What is the division of cytoplasm? You must know that that is cytokinesis. So, in other words, that is the similarities. In both mitosis and meiosis, the cytoplasm divides, and then the nucleus divides at the second point, and then in both, the DNA replication takes place as the third point, and then in both, again, new cells are formed. Already we've got 4 marks. Now, sometimes examiner, like I said, comparison is about the similarities and differences. Now, let us check the differences. Even this one we're going to demonstrate. You may have 2 pages. So let us start by using the first page. But before that, draw a table. And then this table on the left-hand side, write mitosis. And then on the right-hand side, write meiosis. So now let us use the first page to deal with mitosis. So the very same page that we're having, divide it into, or cut it into 2 pieces. Or maybe before that, you need to know where do you think mitosis takes place, and then, or what does it form, what is the main significance of mitosis and also meiosis. Number 1, on the left-hand side, just say it forms body cells, and then these body cells, in brackets, write somatic cells. And then meiosis 1 on the right-hand side, under meiosis, just write it forms sex cells, in brackets, write gametes. Remember, said gametes are sperm cells and egg cells. And then number 2, now we are going to use that strategy that I said we are going to use. Tear that particular page that we are having into 2 pieces. You cut it into 2 pieces. Then the tearing process, remember, it's only one process. So now we are going to say 1 nuclear division takes place. Remember, we are talking about something under mitosis. 1 nuclear division takes place because the tearing process is only one. Now, how many pieces are you having now? You have got 2 pieces there. Then you are going to say 2 daughter cells are formed, and you don't have to cram. Isn't that now you did cut that particular page into 2 pieces? Now, those pieces there are like the daughter cells. 2 daughter cells are formed. And then remember, with mitosis, there's no crossing over, as point number 4. No crossing over. And then remember, crossing over is the one that brings forth variation. And then the fact that there's no crossing over means that the daughter cells in this regard are going to be the same as point number 5. So on the right-hand side, you are going to use another page, the new page, cut it in 2 pieces, and then from there, make it a point that those 2 pieces, you put them together. That was the first tearing process, and then cut those 2 pieces that are linked to each other, and then we divide it into 2 now. Now we're going to end up having 4 pieces there. But now we are going to say the tearing process, remember, it's 2. Now we are going to say 2 nuclear division takes place. And then how many pieces are you having now? You are having 4 pieces now. Then we are going to say 4 daughter cells are formed as point number 3. And then remember here during prophase 1, crossing over takes place. Then you are going to say crossing over takes place. And you also make it a point that you specify that during prophase 1. And then the fact that crossing over takes place means that genetic variation is going to be introduced here. And as a result, the daughter cells are going to be unidentical. So that's point number 5. Daughter cells are unidentical. And then point number 6, you are going to talk about metaphase. Metaphase 1, under mitosis, single chromosomes are arranged along equator. But with meiosis, more especially metaphase 1, chromosomes are arranged in homologous pair along equator. And then with anaphase, under mitosis, chromatids move to the opposite poles. But with meiosis 1, the whole chromosome moves to the opposite pole. Then that is all about this part. Now, let me show you how questions are going to be asked based on the chapter of meiosis. This question is the essay type question from November 2017 question paper. I'm going to read it slowly so that you are able to write it down, because I'm also going to guide you on how to write or to attempt the essay type question so that at least we get total. Write down the following. Say, describe the structure and arrangement of chromosomes. Describe the structure and arrangement of chromosomes, making up the normal human karyotype. Making up the normal human karyotype. Full stop. Also describe the behavior of the chromosomes. Also describe the behavior of chromosomes during the different phases of meiosis 1. During the different phases of meiosis 1. So the most important thing that we're supposed to do here is to underline the key concept first, so that you end up knowing the subtopics of your essay type, so that with regard to synthesis, you get all the marks. Number 1, you are going to describe the structure and the arrangement of chromosomes. You underline structure and arrangement of chromosome. That's going to be our first subtopic. The other one, it says also describe the behavior of the chromosomes. Then you are going to also underline the behavior of chromosomes. Sorry. And then now, with regard to structure and arrangement of chromosomes, you are going to say you don't have to cram. Remember, I told you that in a cell we've got 46 chromosomes. You simply say each chromosome comprises 2 chromatids, then you get a mark for that. Held together by the centromere, you also get a mark. There are 23 chromosomes. 23 chromosomes, 23 pairs. Meaning that we have got 46 chromosomes in a human somatic cells or body cells. You get a mark for 23 pairs and a mark for somatic cells, which are arranged into homologous pairs. You also get a mark for that because I said they are homologous, homo meaning the same. Meaning that similar in length, you get a mark for that, and then they carry genes. I'm talking about this homologous part of it. They're similar in length, you get a mark for that, and then they carry genes of the same characteristics, you also get a mark for that. Then they have alleles of a particular gene at the same loci. Loci, L-O-C-I, is the plural form of locus. Locus is about the position. It's derived from also the Greek word loci. Loci, or they also, and in English we also talk about location. It's just about the position. And the other point is that they have the same centromere position. They have the same centromere position. Then this is what we talk about homologous. Then you also get a mark for that. Each somatic cell has 22 pairs. Remember, this is what I told you last time, to say 22 pairs or 44 autosomes. You get a mark for that. And then the 23rd pair, or you simply say 2 pairs of sex chromosomes, you also get a mark for that. Remember, they form the 23rd pair. And then we also have, you also simply say autosomes are arranged in pairs from largest to smallest. In a karyotype, you also get a mark for that. And then males have XY chromosomes, you get a mark for that. Females have XX chromosome, you also get a mark for that. Then the X chromosome is larger than the Y chromosome, you also get a mark for that. As simple as that. So we have got a lot of marks, and then here they only needed 8 marks. And then the behavior of chromosomes in meiosis 1. Then here you are going to say during prophase 1, you get a mark for that. Chromosomes pair or pair up, you get a mark. Crossing over, exchange genetic material, or crossing over takes place, you get a mark. Between chromatids, you get a mark. During metaphase 1, you get another mark. And then homologous chromosomes, a mark. At the equator, another mark of the cell, another mark. Or you simply say homologous chromosomes. Remember, they are arranged at the equator. You get a mark for homologous chromosome and a mark for equator. In a random way, another mark for that. With the chromosomes attached to the spindle fibers, you need to know this, that the chromosomes are attached to the spindle fibers. You also get a mark for that. During anaphase 1, you also get a mark for that. Chromosomes pair separate, mean that we are not going to talk about chromatids, talk about chromosomes, they separate. And then during telophase, you get a mark for that. The chromosomes reach the poles of the cell. So as simple as that, you get all the marks. Sometimes I told you about the abnormal meiosis. Sometimes examiner may end up saying, explain how the abnormal meiosis may lead to genetic disorder, Down syndrome. Remember, Down syndrome is also known as trisomy 21. Tri meaning 3, and then somy chromosomes. 21 means 21. So in other words, in a cell, you realize that I told you about the sperm cells that is going to, during spermatogenesis, although we're going to discuss, or let me just simplify it like this. Let me say pair 21, they fail to separate, is a gamete, and then pair 21, they fail to separate because we're supposed to have only 23 chromosomes, but pair 21 failed to separate. Then how many chromosomes are we going to have? Obviously, we're going to have 24 because we've got 23 plus 1 of non-separation of pair 21. So now we're going to have 24. That gamete is called abnormal gamete now. Let me say the other side, you realize that the process of division took place, all right, and then there was we're having 23 chromosomes. In other words, that one is called the normal gamete. So now we're going to talk about the chromosome where they fail to separate. Then that process is called non-disjunction. As long as the chromosome fails to separate, we call it non-disjunction. So we are going to say, due to non-disjunction, you get a mark. In meiosis 1, you get a mark. Chromosome pair 21 may not separate. You get a mark for that again. But in meiosis 2, you get a mark for that. You simply say the chromatids of a chromosome 21 may not separate. You also get a mark for that, because you must indicate the difference with regard to meiosis 1 and meiosis 2. And then some gametes will therefore have an extra copy, 2 copies of chromosome 21. This is abnormal gamete. And then if a gamete with 2 copies of chromosome 21 fuses with a normal gamete, that one you remember is abnormal gamete because this has got 24 now. This one, normal gamete, has got 23. The resulting zygote is going to have 47 chromosomes. Why 47? Because it's 24 plus 23. We end up having 47. Then resulting to to Down syndrome. Like I indicated, that's why I said Down syndrome is also regarded as the trisomy 21. So this is very interesting with regard to this one, because examiners sometimes may bring forth a different disorder, Klinefelter syndrome. We've got Down syndrome, we've got Klinefelter syndrome, which has got a similar strategy with regard to this one. But we also have the Hutchinson-Gilford-Progeria syndrome, the Cry-Duchette syndrome, and so on and so on. But for your syllabus, we are going to focus a lot of attention with regard to Down syndrome. And remember, Down syndrome, like I said, is more or less similar to Klinefelter syndrome. Last year, if I'm not mistaken, there was a question that was set based on Klinefelter syndrome. So I'm so excited that at least we managed to complete this. But please make it a point that if ever you've got questions, you must pose them. Next time when we meet, before I start with the other part, I'm going to revise with you those type of questions. Thank you.
All right. Thank you, sir. We have a couple of questions that came through, but of course we are out of time. So I'm guessing you will touch on them maybe next time?
There's no problem. Maybe you could just outline them. I'll touch them. I'll discuss them next time.
We'll make sure that you have the questions so that next time you can...
Please, because last time you said you were going to send, and I did not receive anything, but we need to have them so that before I start with other lessons.
Of course. We'll make it a point.
And thanks.
All right, Mr. Majia, thank you so much for coming through. Life Sciences, Meiosis, man. I would have been an A student.
You think so? You think so? Knowing what you know now.
When I get a sponge, absorb. All right. Thank you so much for coming through. Remember, Sasa will be having these sessions again.
This lesson was brought to you by Kagiso Trust in partnership with the Limpopo Department of Education and Capricorn FM. Kagiso Trust, overcoming poverty. We are saying our goodbyes.
Yeah, all right.